Cells have a life cycle just like we do, starting from when a new cell is formed through cell division and going until the cell either dies or is ready to divide again. Now, biology is a bit odd in this respect. Keep in mind as you're studying cellular reproduction that our cells increase in number by splitting each cell in half. So you go from a parent cell and it divides into two daughter cells. That's just the term that's used. They're referred to as daughter cells. Voila! Now you have these two identical daughter cells, and each daughter cell is identical to its parent as well. There are two main stages to the cell cycle. You have mitosis, when a cell actually divides to make two new daughter cells, and you have interphase. Interphase is in between cell divisions, when a cell is carrying out its normal functions, making proteins, taking in nutrients, secreting molecules, whatever the normal functions of that cell are. The amount of time it takes for a cell to complete one cycle is highly variable. Some cells divide rapidly, like the cells in your bone marrow that make blood cells, or the cells in your digestive tract or skin that are constantly dividing to replace damaged cells. But some cells divide very, very slowly, or even not at all, like neurons in the brain. Most of them don't divide at all, or fully mature muscle cells. The mature cells don't divide at all, although they can be repaired by immature cells that do divide. We'll talk about mitosis, this bit here, in more detail in a separate video. For now, I just want to talk a little bit about interphase, between divisions. Interphase as a whole can be subdivided into three phases. You have the G1 phase, the S phase, and the G2 phase. G stands for growth. So you can think of G1 as the growth 1 phase, and G2 as the growth 2 phase. The G1 phase is where cells are carrying out business as usual. This phase is the really highly variable phase. It can last for hours in rapidly dividing cells, but it can last for days or even months in cells that are dividing more slowly. Cells that are not going to divide enter into a state that's called the G0 phase. In G0, cells are carrying out their normal activities, but they aren't getting ready for cell division. They aren't preparing for this S phase. Sometimes cells that are in the G0 phase, they can be returned to the cell cycle by adding growth factors, proteins that stimulate a cell to divide. So the G0 phase isn't a permanent state, but it takes very specialized conditions and very specialized stimuli to knock a cell out of G0 and put it back on the dividing go-around. Cells that are preparing to divide leave the G1 phase and enter into the S phase. The S stands for synthesis, and specifically DNA synthesis, because the S phase is when a dividing cell makes a copy of its DNA. Cells need to make a copy of their DNA before they're going to divide so that each daughter cell can get an identical copy of the parent cell's DNA. And we'll talk in a later video about exactly how this happens. Understand that DNA replication is absolutely required for cell division. If a cell can't duplicate its DNA successfully, there are mechanisms in the cell that halt the whole process. There are checkpoints where a cell actually will stop if it has not successfully replicated its DNA. Normal cells do not progress into mitosis unless they've successfully made two complete copies of their DNA. Once that has happened, a cell enters into the G2 phase. G2 is a really brief phase where the cell is basically just getting ready for mitosis. It's making the necessary proteins for mitosis and getting them to the right place in the cell. Very brief. This slide shows you both the cartoon version of a cell in interphase on the right and an image from an actual cell on the left. The cell on the left has been treated with molecules to make structures in the cell fluoresce. The DNA is in blue, microtubules, part of the cytoskeleton, are in green, and intermediate filaments are shown in red. Really, the intermediate filaments are just there to show the shape of the cell. There are some key features of interphase to look for. Note that the nucleus is intact. The DNA is still contained within the nuclear envelope. Also note that when you're looking at over here at the actual cell, you can't make out individual chromosomes. You don't see individual long strings of DNA. You just see basically a solid blue field. You can see the microtubules are centered on this one spot here, close to the nucleus. This is where you have the centrosome. And that centrosome is also called the MTOC, for microtubule organizing center. Each centrosome is made up of two centrioles at right angles to each other, like I just sketched there, or you can see them up here. Microtubules in this cell radiate outwards from the centrosomes. You can see there and there. The centrosome actually replicates during interphase as well, so by the end of S phase, the cell has two centrosomes. Non-dividing cells only have one. At the end of interphase, the stage is set for mitosis. Alright, after studying this video, you should be able to answer these questions. Talk about the cell cycle, distinguish between the two major stages of the cell cycle, explain what happens in interphase, and talk a little bit about the G0 stage.